All right, in the last section, you learned about different types of cholesterol and high cholesterol and how it's affected by your diet and lifestyle. Now, unfortunately, cholesterol is limit. It is affected by your diet and lifestyle, but they're finding more and more that it's affected even more by your genetics. So, what we're going to do is looking at Anna's family and seeing if they have one of the genetic diseases that can cause high cholesterol called familial hypercholesterolemia. Um, and we're going to be doing a gel electrophoresis on it, and you guys are going to actually do a real one here. And this is the first of many that we do in this class. So the keywords we're going to look at as allele, you should know what that is from Unit 3. Heterozygous, homozygous, genotype, you should all know what those are. Familial hypercholesterolemia, hypercholesterolemia is I still said it wrong, but that's the we'll talk about more of that in a second. Restriction enzymes are enzymes that cut up the DNA, so we're going to go back to that, but that we talked a little bit about that in unit 1, and we talked a little bit about PCR in unit 1 too, and gel electrophoresis, but we'll review all that real quick. All right, so first, what is familial hypercholesterolemia? Well, as we know, LDL takes cholesterol to the cells for the cholesterol to enter the cells. So just like kind of like insulin, insulin has to bind to a receptor to get that glucose into the cells. Well, so does LDL. LDL has to bind to a receptor. So normally, and this is showing the liver, so it can also take it to the liver. Normally, there's this little receptor on the outside of liver cells. Maybe it looks like this. All right, but and then the LDL will bind to it and will let cholesterol into the cells. But if we have a genetic mutation, that protein will be misformed. All right, as we learned about in sickle cell, the protein might be misformed and it might be like a V there, and now LDL cannot bind to it, so cholesterol can't get in. All right, and then that causes even higher levels of LDL. And as we know, when there's high levels of LDL, that causes plaque buildup or cholesterol to stay in the blood cells. And then that can raise blood pressure and increase our risk for heart disease. So it's all because of that base change that causes the protein to be different, just like in sickle cell with hemoglobin. The hemoglobin changed its shape and then caused it to stick together to do this. This doesn't change the shape to stick together, it just changes the receptor shape so LDL cannot bind to it anymore. Alright, so it doesn't work. Alright, so let's go now and let's review. Well, first let's give a little bit more background here. So FAH is a genetic disease, and it's actually dominant. So you can have, and if I just use F's here, if you have either one of these, you get the FH mutation, so it is dominant. All right. Generally, in sickle cell, we looked at recessive, but this is normal in this case. All right, so you just have to flip your mindset around a little bit that these two people are dominant and have the disease. The dominant have the disease, whereas the recessive is normal. So it's just passed on, going to be passed on a little bit differently than the sickle cell was. Alright, so let's review gel electrophoresis. So the first thing we do is we take the DNA from the patient, so we're going to take it from Anna's family. Then we use PCR to make lots of copies. Alright, so PCR, because we can't just take one little copy of one little thing of DNA and be able to look at it. So we have to make millions of copies. So PCR makes millions of DNA. Alright, then restriction enzymes will cut up that DNA. So that's what we're seeing over here. So we take the gene, we absorb it, and then we take the gene of interest and we'll cut it. And depending on the bases, we'll cut it into different lengths. So restriction enzymes cut at one segment. So if they cut at GGAA, All right, so one person might have GGAA, might have just one of these sequences, whereas another person might have two. All right, if they have one, it will cut once, and then you'll have two bands, one on either side, 1, 2. And then if this person has GAA twice in their DNA, they will now have three bands, 1, 2, 3. All right, and then they show up as different numbers on the gel electrophoresis. So the gel, remember, separates the DNA by size. The shorter ones go to the bottom, and the longer ones stay at the top. And you're going to then compare the DNA to known samples to see who has the FH mutation. Remember, you just compare those bands. All right, so we're going to look at Anna's family again, and we're going to see who has this FH mutation. So your goal is to figure out how to complete this gel electrophoresis, analyze it, and figure out who in the family has a mutation, and again make another pedigree. So this is kind of very similar to the sickle cell. We're just going to be doing with the FH mutation.